Good afternoon and welcome to the third interim symposium session of the International HLA workshop. Today we have two speakers. We start with Nico Vins, who is working at the University of Nantes, and together with Pierre-Antoine Gros, he's leading the SNP HLA project. So Nicholas, please go ahead.
Well, hello, everyone. I will give a, a short introduction of our project, the SHLAC, and then I will give the floor to my PhD student, Venceslas Douillard, who will show you the results we obtained with this project. So the SNP HLA Reference Consortium that we call the SHLAC. HLA is very associated with a lot of diseases in traits. Around 25% of all GWAS catalog diseases and traits present at least one associated SNP within the MHC. This is what we see here. This is the number of significant association per region of the genome from the chromosome 1 to the chromosome 22. And what we see, the large peak in the middle is the HLA region. The problem with GWAS is that it associates a SNP or a region, so here the HLA region, to a disease or a trait. And we don't know exactly what's going on there. So what we want to do is go further from the SNPs. And to do that, we need to do HLA imputation. HLA imputation is taking the SNPs we have from the genotype people, compare it to a reference panel with the SNP and HLA to obtain the HLA alleles. The objectives of our consortium, the SHLAC consortium, is to get more data, quantity, and diversity to improve the HLA imputation. A lot of the data available right now are from European ancestry, and we need more diversity to obtain better HLA imputation from underrepresented population. The second objective we want to do is to improve HLA imputation by using the already existing techniques that we have, such as HIBAG or SNP2HLA or HLA-IMP. And maybe we didn't stop that yet, and if anybody is interested in doing that, we would be happy to share some of our data, develop new HLA imputation techniques. And what we are doing right now is trying to make customized reference panel to better impute HLA. The third objective we want to do is to create a website that would be accessible to everybody, so anybody could impute its HLA from its own data, and to have a customized reference panel to better impute its HLA. This network is now has 65 participants from 32 teams across 16 countries. Today, I invited two persons to talk in the parallel session right after this session, Eric Castelli from Brazil and Jérémy Decouchant from, whom is now from Netherlands. He was from Luxembourg since recently. Also, we have two large data sets that we are collecting right now. A data set from Finland that I have to write the paperwork, and I still didn't do that, and it's a shame on me, but I will do that soon. And another cohort of around 500 individuals from Benin, and we will do the HLA typing. The genotyping is already available. We had a small setback with this project. We obtained money in 2019, but due to political reason, local political reason, the money was frozen since then, and the money should be back on track by the end of this year. So with that, I just wanted to give you a short introduction of the Schlag, and I want to thank the whole Team 5 here, which is collectively working on this project. And now I will give the floor to Venceslas, who will be presenting our results. And this is all right. How do you do that? Do you see the new screen?
Not yet.
Not yet. Let's see. Yeah.
Yes.
All right. There we are.
Hello everyone, my name is Venceslas Douillard and I'm a PhD student in my second year. I work with Nicolas on the matter of HLA imputation, but more precisely, the building of reference panels for admixed population and overall the impact of diversity on HLA imputation. In the past few years, as Nicolas said, there has been an overwhelming number of SNP associations in the MHC region. This proves that the genes in this region, whether they're HLA or not, are really important in diseases, whether they are autoimmune or infectious diseases. The problem is those kind of associations do not pinpoint a specific HLA allele and are difficult to interpret biologically. The development of sequencing technologies allow us to do these associations directly on HLA alleles, but it is still hindered by the fact that it is quite costly and it needs a little bit of expertise. In parallel of the development of these technologies, there's also statistical inference of HLA genotypes that have been developed. This inference allows to use SNPs, so small polymorphism on the DNA of people, to infer the HLA genotypes, but you need a reference panel. And the reference panel direct what you will have as a HLA imputation. So briefly, what is a reference panel? It's only a data set of individuals for which you have SNP information and HLA genotype information. With an algorithm of your choice, Nicolas mentioned IHIW because this is the one that has been independently proven as one of the most accurate. You create links between the SNPs an individual has and the HLA genotypes. So you have a reference panel. Then, with only SNPs, you can predict the new HLA genotypes. With IHIW, for example, a model, a reference panel could be looking something like this. So SNP haplotypes with an HLA allele and the probability to have these two at the same time. So quite simply, if you have this SNP haplotype, you will choose the most probable HLA allele. The thing is, even if HLA imputation may seem simple like this, there are still huge challenges in HLA imputation. First of all, sorry, just I will do this. First of all, there are no gold standard in HLA imputation from SNP genotypes. Researchers are still arguing whether they should use a reference panel that is specific, meaning that you should really create your model, your reference panel with people from one population, whether it is Japanese population or Finnish population. And others argue that you should go with more multi-ethnic data, so creating reference panel with everyone. The way to measure the accuracy of HLA imputation is also debatable because accuracy is often used, but a specific allele which is unpredicted 20 times in a 1,000 individuals still has a 99% accuracy. So it can be really misleading when you interpret the results. Sorry, just a technical problem.
Okay.
Yes. Nope. Yes. Sorry. And the other challenges in HLA imputation is that even though HLA imputation is not really computer demanding, the creation of such reference panels can need months of calculation on a personal computer. We had the chance here in Nantes to work with 3 supercomputers, and mostly the one in the University of Nantes, which allows us to create reference panels of thousands of individuals and thousands of SNPs in 24 hours for the 5 main classical HLA genes, A, B, C, DRB1, DQB1. And we are willing to share our infrastructure to help the creation of many reference panels. To investigate the impact of the diversity in HL data on HL imputation, we work mostly with 2 datasets. The 1000 genome dataset, which is known to be really diverse with 26 populations across the world, and the Kappa dataset, which is a smaller dataset, but still almost 1,000 individuals who are admixed European African. Our first results really focused on the 1000 genome population because it is the most diverse. So in abscissa here, you can see the 26 different populations that we have in the dataset and the 5 HLA gene. What we've tried to do is to create a reference panel of every population except one. Then we try to impute the HLA of the left out population just to see how much a population can stand out from the other and impair HLA imputation. What is really striking here is with the Mexican and Peruvian population, the HLA imputation accuracy is not that great. And it comes from a really simple thing, as HLA experts may know, is that HLA alleles may be specific to certain populations. So if your multi-ethnic model has a lot of individuals, but not the one from said population, you will not be able to predict accurately those populations. If we just remove the American superpopulation just to see a bit more clearly, we can see that even if we're not talking about specific alleles, the difference in frequencies lead to changes of imputation in other populations. As in the Kenyans here or the Japanese population, you can see that overall their individual accuracy of HLA imputation is lower than other populations because the difference in frequency impair HLA imputation. And another result that is quite expected is that B and DRB1 HLA genes are the harder to predict because they have higher polymorphism rate in those HLA genes. What we wanted to do, we wanted to move from the multi-ethnic reference panels and try to think about how to create a really specific reference panel. What is often done in population genetics is to use PCA, a dimension reduction method, to visualize the ancestry of a population based on the SNPs. What we thought we could do is to do this dimension reduction technique and select from the population we're trying to impute the population that we have in reference that are closer with the overall genetic similarity. So basically, when we reduce dimensions, we have the SNPs from individuals, coded 0, 1, 2 if they have polymorphism 0 once or twice. And we obtain something like this with 2 axes. We used the SNPs in the MHC region, and we did not have good results with PCA. It really was just a scatter plot with no distinct ancestry. But we used another technique, which is called UMAP, which allows us only from the MHC region to see the difference of ancestry. And what is really interesting is that with our population we're trying to impute, or admixed population, here in black, we do not have a population of African American as a whole. We have 2 distinct parts. One that is more closely related to African populations of the 1000 genome dataset, so Kenyans here or others, and another part which is more likely to be closer to European genetically. So once we saw this, we wondered maybe we can automatically detect those groups, those ancestry groups in an admixed population and create two different models for one population, for the kappa population. So once we can do that, we can evaluate, is it a good thing and does it bring something to HLA imputation? To give an overview, we made a protocol to evaluate this, which worked with the two datasets that we have. The 1000 genome, which is used for the training, and the kappa, which is used for validating our model. So from the 1000 genome, we create models from 200 individuals picked either from one of these superpopulation, and we compare it among them, but also against 200 individuals from the whole 1000 genome dataset chosen randomly to see if the multi-ethnicity is better than using a specific population using a label. Then we're also comparing this to 200 individuals not chosen because of a label like African, American, East Asian, but because they are closer genetically shown by PCA or UMAP. We use the two techniques. And then we're seeing if we impute correctly the kappa data. For now, we have only analyzed the results for this part of the protocol, but we already have this, and the new results shall come soon. To give you a little bit of preliminary results, for HLA-B, what is really interesting to see is that here in abscissa, we have the different population I was talking about in the last slide, and some alleles of the kappa population are not predicted by any other models. And what you could say is that maybe the HLA allele is not present in our original data, but it's not always the case. Sometimes it is only in the original data, but the algorithm can't make the link between the two. What is also really interesting is that if you compare 200 individual 1000 genome datasets, well, you have lower imputation results than if you take a more closely related population like the African population. So it is very interesting to see that specific and customized reference panel may be the solution here. Even if you look closely, using the whole 1000 genome dataset, so 2000 of individuals, you have better results. So maybe if you have a limited number of individuals, you should go to specific reference panels. But if you have enough data, maybe it should be a more good idea to go to multi-ethnic panels. We also have new interesting algorithms that are published or ongoing. I'm thinking about Cook HLA that has been published recently, or the Deep HLA that we will be looking into in the future. And the results on the specific models with the UMAP and PCA should come soon. I would like to thank the whole team again for their help, and Nicola especially, and thank you for your attention.
Thank you. So we have some time for questions. Maybe I try to get one question here. Is from Marcelo Fernandez Vinha. Maybe I'm not sure whether you can connect in, Marcelo, yourself, but otherwise I will ask the question. Have you tested the IHIS cell panel that is available from Seattle and that has been typed for classical HLA genes by NGS?
No, we did not try that. I did not check this particular data set. I'm not sure if it was sequence on the whole region or on specifically on the genes. The technique really require to have SNPs not only very close to each of the HLA genes, but also 500 kb before and after each of the genes we want to impute. So I will try to see if these data are available and see if we can use that, of course.
Yeah, because from the HLA side, these are part of the workshop for many, many years and have been typed extensively. I think most of them are even part of the QC project by Tammy. So there's a lot of knowledge on the HLA part, at least. Having said it, from my side, if you would do this on EBV LCL and these SNPs, is there anything known about mutations that occur during culturing cells? Do you need to do this on uncultured and manipulated cells, or can this also be done on EBV LCL? What is the noise you get if you use these EBV LCL?
Yeah, that's a good question. As we are using a large population, I mean, we really need to have at least 100 individuals in a reference panel to get good results. So we will always impute better the high frequency alleles, and the challenge will always remain for the low frequency alleles. So if a mutation occurs, it will create a rare new haplotype that we will not be able to really see or even impute in the with the reference panel in the data we want to impute.
Okay, then there is a remark from Steve Marsh saying there is no A7040 allele under any predictions. Maybe.
Oh, so in my in my slide, you mean?
I don't know. Steve just posted this statement here.
Okay.
He says yes.
Yeah. I did not check for the number of HLA allele here. I wrote one, but yeah, I did not check that.
Okay.
Also, HLA-B should be HLA-B. Yeah, I think Steve is pointing at some structural nomenclature issues.
Okay.
Then there's a question from Makanga. What data is to be submitted for this project? In what format? Any sample size defined for a specific population that needs to be included for analysis?
Yeah, good question. So what we really need is both SNP data and HLA allele data. That's really required. And I would say around 100 individuals per population is probably somewhere we can start with to have a big enough reference panel to be able to do any a prediction of the imputation of HLA in the second data set.
Question, Marcelo. May I ask a verbal one? Paul Norman is running a sequencing of full HLA haplotypes. Can you pair this effort with Paul Norman? So really you'll get insight of these haplotypes that have not been characterized.
Yeah, we can try. I saw his talk 2 days ago, I think it was, and it looked really interesting. What I'm wondering is the frequency of these haplotypes, because as I was saying, the imputation will only work if it's frequent enough, and we will always get a really hard challenge for low frequency alleles and low frequency SNPs as well. The goal is to have large data set that we can impute only with the few number of SNPs. Few is relative. It means between 10,000 to 50,000 in the whole MHC region, which is what the DNA chip are giving.
We got a freeze on our end, Nicholas. Apparently. I think this is a sort of technical issue. That there is a follow-up session after this plenary session. So if there are additional questions, I think we should forward them to the parallel session after this session. See, something seems to move again. Well, no, not really. I think we better move forward with the next speaker, which is Saimak Bakram, who will explain a little bit more about the COVID HLA genome project within the context of the international workshop. I see here, I will give you the floor, Saimak, in a second. You should be able to share your presentation right now. And because, well, you're in twice, so I'm not sure whether, yeah, there we are, good.
Do you see my screen?
Not at the moment. Now we do. Yeah.
I can't see it myself.
Yeah, but you're good to go now. Yeah.
Okay. Sorry. You can hear me and you can see my slide deck?
Yes.
Yes, we can.
Okay. Okay. Thanks very much for the opportunity to present the status of our project, COVID and HLA. I will rather briefly present the project and where we are at, and we can have discussions, and if there are groups who will still want to join us. So this project was initiated in Strasbourg, myself and Rafael Carapito, who is also on the call, and the abbreviation for collage COVID HLA genome. So I'm not going to make, of course, an introduction to COVID. I think everyone knows what we are talking about. Won't make a long introduction on HLA either, but for the youngest among us, students, if they are there, everyone should realize that the MHC, at least in the mouse and guinea pig, was identified because of its control of immune response. So immune response in particular to infectious agents, and throughout the years there has been major work with regards to MHC association and infectious diseases, although the results are not as impressive as MHC association with autoimmune diseases, mainly because diseases and infections have had the time to fashion by natural selection the MHC alleles. COVID-19 will be an opportunity to catch a disease at its inception, at least the SARS-CoV-2, and this is the ultimate aim of the project. As all of you know, we are not equal in front of a SARS-CoV-2 infection, even if you harmonize cohorts of individuals for age, sex, comorbidities, etc. You have, let's say, a decent portion of individuals, up to half, who would remain asymptomatic. A small portion, small or rather large, small portion would be symptomatic, but with manageable symptoms, influenza-like symptoms. You have approximately 15% of patients who go into pneumonia, who will need supplemental oxygen, and a small fraction of patients who end up in the ICU with ARDS. And of course, these are the fraction of patients who are responsible for the severity and the death toll of the disease. Of course, one of the clear culprits are HLA genes, and the aim of our study is to understand the extent to which MHC alleles are responsible for susceptibility to infection and to various clinical course of the disease. So the aim and rationale of the study is quite simple. To establish the extent of association between mainly HLA, but also MYC genes and KIR loci with COVID-19 susceptibility, infection severity, and ultimately response to vaccine. Because you are also aware of the fact that, like all vaccines, not everyone responds totally to a vaccine. The best vaccines are supposedly have good response rates of 90%, but some of them are around 70%. So there is a fraction of individuals between 10 to 30% of population which does not respond to vaccines. And this might be ultimately interesting to understand why. And the other point which will be interesting is to basically study the antiviral immunity, which are mediated by, as you know, T cells and against viral peptides presented by MHC class 1 molecules. So in order to do as good as a study as possible, we have to eliminate the confounding factors, which are age, because you know the age is one of the key factors for severity of COVID-19, as is sex and as is comorbidities, and of course the virus also intervenes in this the pathology, especially with the advent of various variants who are running across the planet. In this study, we only focus on HLA, KIR, and MYC, not in the genome in total. There are other works, including by us, on whole genome sequencing of COVID patients, but it's totally independent from the study I am presenting to you today. So to this audience, I shouldn't mention this, but there are already some preliminary information with regards to the genetic influence of the host on COVID-19. There has been several GWAS studies which have been published, and there are genome sequencing, exome sequencing studies who have also been published who have hinted to, for instance, the innate immune genes. Interestingly or uninterestingly, the exome studies and the GWAS studies, as usual, don't always overlap, but that's basically a fact in any GWAS versus whole genome study. There has been, as of today, a number of reports with regards to HLA and COVID from different parts of the world. As you see, Europe, China, Asia, etc. One thing is common is that these are mainly small cohorts. So the highest one is up to 300 individuals. And most of them basically make a common bag of COVID patients. So they only include COVID patients without going into detailed stratification. Our study is based on 4 categories of individuals and patients, which is basically their real life status of COVID in the human population. So the first group, G1, the asymptomatic individuals. The second group, G2, are individuals who have only flu-like symptoms. Third group, individuals who need to go to the hospital and be hospitalized because of supplemental oxygen requirement. And of course, the fourth group are patients who have ARDS in ICU and are under mechanical ventilation. And of course, these groups do not communicate to each other. For instance, if someone from G3 goes to G4, he will be regarded as G4. From G2 to G3, we include him as G3. So it's always the higher group which is necessary. For group 1, in order to ascertain infection, we either need, of course, DNA validation, qRT-PCR of the virus, or serology. But for G2, 3, and 4, we definitely need viral genome positiveness documented. We have we will do two different analyses. Basically, one of them is where everyone in each group is under 50 years of age. Because why 50 years of age? Because that's where the curve tries it's changed direction and the mortality goes higher. Actually, it's over 60, but 50 is a very stringent age without comorbidities. So we have at our final analysis, we do an analysis with individuals under 50 without comorbidities, or an analysis with the entire patient groups, irrespective of age and comorbidities. These are the clinical and biological data that we have included in the study, and whoever is interested to participate, we need to have this information from your cohorts, so sample ID. Of course, everything is anonymized by the group who sends the data. Sex, age, BMI, ethnicity, if it's available, co-known morbidities, ARDS, yes, no, SARS-CoV-2 PCR, and the immunization, and basically the information in which group the individual belongs to. These are the sample requirements for analysis: 3 to 5 micrograms of high molecular weight genomic DNA. The analysis which will be performed is high resolution HLA-KIR and MYC typing by NGS. So for HLA loci, all 11 loci are analyzed to 2 fields resolution, and we cover the cost for the sequencing if it's done by us centrally. If a group wants to provide his or her own sequencing data, of course it should match ours, but we cannot cover cost inside participating lab. If the samples are sent to us, we cover all the costs. And this is basically the analysis will be done. The study would be between specific HLA, KIR, MYC types, and SARS-CoV-2 infection and clinical manifestations of COVID-19. So where are we at now? This is a schematic of the groups who have already participated in this work. So it goes from France, Australia, Hong Kong, Middle East, and Europe. We have no participation from sub-Saharan Africa, and especially from China. But other than that, we start having a lot of interest from these centers. As of today, we have a bit over 3,000 samples who have been collected. This is a breakdown by the country, the city, and the center, so it's as precise as it could get. As you will see, almost half samples are French, and the others are splashed across different countries and continents. And we have good hope to obtain within the next 2 to 3 weeks to basically double the size of the cohort, 500 plus samples from France and 2,500 from the UK. This is important breakdown by severity groups. From the beginning, we knew that this will be the G1 will be the most difficult group to have, because these by definition do not go through the hospital system, and all our participating centers are basically clinical groups. So by definition, we don't have access to the G1. But other than that, it's yeah, there is a good representation of G2, G3, and G4, and approximately a quarter of the samples are unclassified. Here we go basically at the same stage as what has been published with COVID, which basically include COVID patients. But so this is with regards to the criteria I mentioned. The strict criteria are patients under 50, no comorbidities, and the extended criteria are all patients, individuals. And you see that, of course, the restricted cohort is smaller, but still not bad in certain parts. And basically, nobody has ever done this to be so strict. But I hope that this will be useful at the stage of the final analysis. This is, again, the situation as of today. Close to 2,000 individuals have been fully typed. 1,200 will be finished in the next, let's say, less than 2 weeks. And depending on the additional samples who are already en route, we will decide at one point, which will be not far away, to basically put a cutoff on the study and start the analysis part. This is something which is available on the IHIW website. I lead the project with Rafael, who is in the lab, and who most of all the people who have interacted with us know Rafael. We have an international steering committee with members from NMDP, FB, JSHI, and of course Eric from the workshop, and several other scientists, and an international scientific advisory boards who covers basically whether immunology, MHC, and some of these people are actively involved in COVID research, whether by antibodies or by HLA. Further information is available here. And having said that, I am looking forward to discuss with the audience.
Thank you, Saimak, for this presentation. There are a few questions coming in. The first one is from Franz Klaas. I tried to unmute him, but technically something goes wrong here. The question is, why do you not include ABO blood group as well? Associations have already been demonstrated and can interfere.
That's a good point. At one point, we may sequence the samples for ABO as well. Yeah.
Because you're doing your typings via DKMS, right?
DKMS or histogenetics. Yeah.
Yeah. So DKMS will be able to do that. Yeah. Then Dominique had a few questions. I'm not sure whether Dominique can bring them in himself. Otherwise, I will read them. So Dominique suggests to add a seasonal factor, winter, summer climate data, which is easily available. Is that something you can work with?
You mean seasonal data based? I don't think the cohort is that huge for involving other factors as well. But that's yeah, that's something we will involve in the ultimate analysis for the workshop, of course. Yeah, good point.
Dominique, you had another question or comment?
No,
Right?
it's not a question. It's just that I will contact the Chinese and the Greeks and the Egyptians that have already expressed their interest. So now that there is clearly a study that can be presented to them, they will join probably.
Okay, great.
And there's Frauke Degenhardt, who's having a question.
I'm unmuted. Maybe I posed it myself. It's a rather long question. Hi, Frauke Degenhardt from Kiel. I work with HLA and also involved in one of the first genetics analysis of COVID. And just my comment is that there have been very diverse reports on the HLA, I know. And the largest genetic analysis by the HGI consortium, I don't know if you're aware of that, they have more than 36,000 samples, do not show a signal in the HLA. And also our own study based on HLA imputation did not show anything there in susceptibility and severity. I guess you're probably aware of that. And we are wondering here if maybe then the rare variants should more play a role if we cannot observe them in the genetics. So we would expect to see also something here if there were some very common variants associated with COVID-19.
What do you mean rare? You mean rare alleles or
rare HLA? Yeah, rare HLA alleles. Yeah.
Yeah, I'm of course I'm aware of the data, but as I mentioned, basically it's they pool COVID patients, so they don't stratify as much as we do. So yeah, that's what is what's the difference between us and them. But I am whatever the date, as one knows, it's whatever the date takes you to. So
yeah. I'm just saying, also stratified analysis from our part do not show any signal. Just if you want to get in contact here, we're happy to discuss that further.
Okay, could you send me an email, because yeah, sure. I was already, yeah, okay, if you can send me an email.
Yeah, I will make a screenshot right now.
Can I, this is Steve Marsh here, Eric, can I just come in to say that that was essentially what I wanted to ask, is that there are clearly a number of publications now that are coming out looking at HLA and COVID, and that, you know, we published a number of those in HLA, and there's no consistency in the reports. There's no smoking gun really there. What you tend to see is, I think, in differences in populations, you're finding more frequent alleles in some populations being the markers. And I'm interested in this. We're been looking at doing something here in London as well with our nearly 800,000 donors. But I'm not seeing that consistency of data coming out of HLA, of markers within HLA, and therefore I am a little bit concerned that we might not see what we all are expecting to see.
Yeah.
Well, welcome to HLA Disease Association.
Yeah, this is not an area that I usually spend a lot of time in.
Yeah, me neither. So
I think this also more or less answers the questions from Marijke Wendorf. How reliable do you think are the so far reported HLA associations? It seems like there is hardly any overlap between different studies on the allele level. So that's basically what you say, right?
Again, yeah, welcome to HLA Disease Association. That's the problem with, yeah, there is 50 years of literature on HLA disease, and except for very few, especially in infectious diseases, it's very difficult to replicate. But yeah, I mean, here we have gone through utmost pain to eliminate co-founding factors by going under 50, etc., etc. yeah, it's like GVAS. It's it's a difficult thing, but it's very clear that, I mean, basic 101 immunology, the ultimate, the way the virus is eliminated, unlike what you hear every day on the TV, is not by antibodies, it's by T cells. And at one point, there will be an issue how the vaccines work, and what is the epitope, and etc. Especially for next generation vaccines, which will be much broader. And I think we have a shot at it, because otherwise, what we have learned in immunology doesn't make sense. But we need very large cohorts, and especially clinically annotated cohorts. You just put the entire COVID in one bag, I'm sure you'll find very different things.
There's a question from Saxon Core, Charles. Are you able to ask it yourself? I try to unmute you. If not, I will read it out. Would it be possible for us to provide 4-field resolution HLA typing data of Japanese COVID-19 for the components, but we are lacking the KIR typing data?
Yes. The KIR typing, actually, as you know, it's a very contentious issue. Which KIR you type, etc. So if you can contribute the HLA, that would be great from Japanese. And we have had much interest from Japan, but not yet any data contributed to the work.
And then we have a question from Eric Castelli, Brazil. Does collage accept data from samples with whole genome sequencing and HLA alleles called directly from the sequencing data?
Yeah, if you can impute to 4-digit levels, yes, we can include it.
And to follow up on that, Steve Mack asked, what type of KIR typing is being done, high resolution or presence absence?
A mixture of both. Basically, is presence absence. Yeah, basically, yeah.
Johanna Schetterlich can share data from the German cohort with you, so please note that. Then Juliana Cardozo, is there any intention of comparing these data with individuals negative for COVID-19?
That's a big question in any infectious disease. So if the person who is negative because he did not come in contact with the virus, or he or she did come in contact with the virus and remain negative, that's something that you can never prove. So I think negative people, there will be, we cannot, to me at this stage, we haven't thought about including it, because what is the proof of negativity?
Sure, yeah. And then there is a comment from Esteban Arieta Bolianos. In addition to Steve's comment, I was wondering if clinical classification can significantly vary across countries, for example, the decision to hospitalize a patient. In view of this, would it be possible to obtain a more controlled outcome? For example, would it be possible to obtain the CT values of a subset of patients and see if there is a correlation with viral titers that could correlate with clinical outcome? So more objective classification.
First of all, to my knowledge, there is no direct relationship between the CT and severity, and that's an information that we have not integrated. But basically, in all countries that we have included the clinical, I mean, if patient needs oxygen, basically he or she mainly has to go to the hospital. And the ICU, I mean, there is no way that in different countries someone would be in mechanical ventilation in a conventional hospital ward. So yeah, it's not perfect, but it's as good as we have been able to get.
And then there's a question from Attila Berses. are you studying severe cases with autoimmune components?
With autoimmune components?
Yes.
What do you mean by autoimmune components?
Let's see if I can bring in Attilio himself.
Hi. So there was a recent Nature paper suggesting that autoimmunity plays a very significant role in severe COVID, and this is, what I'm referring to. Whether you have some of these severe manifestations where you would be able to identify those which are suspected to have some autoimmune component.
So by autoimmunity, you mean autoantibodies, for instance, to interferon, etc. No?
Yes.
Okay. Now we haven't been testing autoantibodies, but according to the publications, it's around 10% of severe patients. So it's not, but we haven't factored that in. No.
Because I think if we want to find something where HLA is important, we have to look at some very specific and on those areas where we have the highest chances of finding something, and that's why I thought where antibodies play a role, this might be something that might also give a little bit more chance. And I have a second question, which is relates to long COVID, because long COVID is again only a small portion of the patients will develop, or well, not very small, but it's a smaller portion. So that's another area where do you plan to study long COVID?
On principle, yes, but at least to me, I mean, there is no, as of today, to my knowledge, there is no clear definition of what is long COVID. So it's obviously an issue. I think once the pandemic will be over, it's going to be the major issue which will remain. For instance, in Strasbourg, we are setting up a clinic only for long COVID. I mean, there are other cities, I saw in Stanford, etc. But it's very difficult to identify what is the long COVID. So yeah, eventually yes, but eventually yes.
Thank you, Saimak. I think we need to.
Can I just ask one last question? It's Steve Marsh again.
Very short.
Sorry, I'll be very short. Given that you're stratifying into your G1, G2, G3, and G4, and I'm assuming that most of the samples that people are going to be providing, which are coming through hospital contacts, are going to be from G3 and G4 patients, and you're going to bias your analysis to that end. How do you propose to get enough G2s and certainly the asymptomatic, the G1s? How do you get that? Because in one sense, what the markers that we're looking for here, it may be you need more of those to to elucidate what's actually taking you to that more severe disease.
Okay. Yeah, good point, Steve. But actually, the group we have leased from is not is actually G4, G1, and G4. So we have enough of. I mean, we have a large amount, comparatively large amount of G2 and G3. This is something that unfortunately we cannot control what people send us. The lack of G1 is a fact. We might be able to do this through basically centers who do large-scale screening, but it's something we are working in. Yeah, I think every point that I heard today it makes sense. The question is, in practical terms, what we do. So the autoantibodies, the fact is that the groups that send us, they don't have sera. So yeah, so basically, if you go through HLA labs, a lot of them don't collect sera for these patients. So these are all very practical issues which do not allow to do everything we want.
Good luck.
Thanks, Emma. We really need to conclude this session because we have parallel sessions that are about to start. Thank you all for your contributions. We have 3 parallel sessions that will start in a few seconds. One on the SNP HLA reference consortium, another one on the population genetics, and another one on the HLA immunogenetics COVID consortium. So if you have subscribed, you have received links to these sessions, and please proceed to the Zoom meetings for that. Thank you for now, and we'll see you later. Bye-bye.